I work here at the Queen's Library. I've been hired very recently to do what's called green the Queen's Library. So that's our logo down here on the bottom, and I am the compost assistant. This program is funded by the Greening Western Plains Fund. So we're in our final year of funding for that program. It's a 3-year grant process. We're hoping to find funding to continue this sort of programming after this year is over. This is one of the many things we do in the greening of the Queen's libraries. This workshop, as well as we have compost collections at multiple libraries. We had compost collections downstairs on the sidewalk this morning from 10 until noon. 8 until 10, right here at the Steinway branch library every Monday. And then we have compost collection at the Sunnyside branch library from 10:30 until noon on Saturdays. Broadway branch library from 1 until 3 on Saturdays. The Woodside branch library from 5:15 to 6:30 on Tuesdays. And we hope to expand to the Long Island City branch probably at the beginning of next month. We also have environmental documentary film screenings, and as well as programming like this. I'm going to be teaching folks how to build their own window farms, which is aquaponic systems that you put in your windows, and you can grow them out of like recycled or upcycled water bottles. You have to get a little water pump. We're going to be giving one of those away whenever we build it. That's going to be next month, probably at this branch and the Sunnyside branch. We're going to be teaching this class again at the Sunnyside branch on the 25th, I believe, Monday at 6:30. So I encourage all of you to sign up for our newsletter and find out about all of those classes. We also have a garden back here in the back. We have volunteer opportunities to work in the garden with a variety of other people. This email sign-up list is also going to be your key to entering the raffle for the mushroom giveaways that we're going to have later on whenever we wrap things up. So I'm going to talk a little bit about fungi broadly, and then we're going to jump right into how to grow edible mushrooms. So if you wouldn't mind hitting, actually we can't hit the lights because they're in the closet in the back. So I hope, can everybody see this board? Oh, okay? Alright. So fungi are in a completely separate kingdom. It's the fungi kingdom. They're not plants, they're not animals. They're completely different. Just to give you a broad understanding, I'm not going to go too depth into any of this stuff right now. The purpose of this workshop is to learn how to grow these. And that said, I am not the foremost expert. I'm not a mycologist necessarily. I've done a bit of research in order to present the basics to you today, and I hope that it can, just plant a seed in your mind so that you can feel empowered to go and experiment yourself. So fungi are its own kingdom, and there's a lot of different types of fungi. Most of them are many-celled organisms. They have strands of single-cell hyphae that goes underground, and it often decomposes different materials. There is also, a bunch of people know a bunch of different kinds of mushrooms. There's lichen, there's yeast mushrooms, bread molds. Those are just a few examples of the many different kinds of fungi. As well as rusts and some other common forms that you may or may not be aware of. So there's 3 major types of mushrooms. When people refer to mushroom instead of fungi, they're referring to 3 major types. There's mycorrhizal, parasitic, and saprophytic. So those 3 I'm going to talk about very quickly, and then we're going to talk about saprophytic a little bit more in depth. So mycorrhiza is a symbiotic association between a fungus and the roots of a vascular plant. Generally, this is a mutualistic relationship that can occasionally become weakly pathogenic, which for the most part, the mycorrhiza is Oftentimes, gardeners use it when they're inoculating seeds to plant in their soil, so that the mycorrhiza can go deeper down into the soil and have a beneficial relationship with their roots, creating larger networks, as you see over here, in which it breaks down elements and nutrients in the soil and makes it bioavailable for the seedling to take up into its root system. And it also takes some nutrients and some sugars from the root system of the plant, and it kind of acts as the forest's internet, the web of the forest, kind of like redistributing nutrients and different minerals where it's needed to other plants. There's actually been studies showing that plants can grow in dense understories of canopy trees even without all that sunlight, because it shows that the mycorrhizal networks are bringing sugars from the outside from the outer edges of the forest into the forest in order to feed those smaller plants and trees coming up in the center of the forest and create a more dynamic ecology.
Yeah.
So,
do you want all of them out?
No, this is fine. We're only going to do slideshows for a little while.
Okay, I can give you one more. I think I can get the front.
Cool. So, parasitic fungi are members of the fungi kingdom as well, which thrive on latching onto other organisms and taking nutrients from them. They're types of symbiotic fungi, but they are not multi-mutualistic, because they don't tend to give anything back in terms of added value to their host in return for sustenance. So, one example is the fungi that kind of feeds on small insects, such as ants and wasps and spiders. It actually gets into the body of the insect through the cracks in the exoskeleton, and the mycelium or the hyphae run through the body. It sort of takes over, and it could be called a zombie fungi, because it kind of takes over the brain. It tells the insect to go to a place where it's beneficial for the mushroom to sprout and sporulate, so that its spores can be distributed throughout the forest. The other type of parasitic fungi is the type that grows on trees and other woody plants. And the reason that it's parasitic as opposed to the other mushroom that is good is when it grows on these trees, the trees are actually still alive and thriving. There's usually not too much wrong with the tree. So it actually acts to break down the tree as the tree is living. Now, some of these parasitic mushrooms have been shown to be actually beneficial in thinning out forests of the weaker trees that are actually still alive, but they're still considered parasitic fungi. So the saprophytic fungi is what we're going to be talking about today, because we're going to be growing oyster mushrooms. And they feed on dead plant material and animal remains. Many are extremely beneficial, breaking down this organic material into humus, materials, minerals, and nutrients that can be utilized by plants. Without these, fungi would also disappear under a mountain of unrotted leaves and logs. So these are really important. These saprophytic fungi are very important in maintaining soil health throughout all ecologies. Some would say that a forest is not possible. A forest is not truly possible without mushroom. So I know a lot of people think of forests, they think of the big trees, they think of the understory, and maybe some ground cover, even vining plants, maybe even root crops if you're kind of, thinking a little bit deeper. But in reality, you really have to have fungi. They're kind of the first. There's 3 different types of saprophytic fungi. There's the primary, the secondary, and the tertiary. So the primary breaks up larger chunks and larger mineral chains, chemical chains, and it goes on down until things are really finely broken down into bioavailable nutrients that roots can then uptake again. Now, fungi is not the only one that does this. There's also, a litany of bacteria and, earthworms and other insects and all sorts of things that break things down in the forest. But without that initial, especially the primary fungi decomposers, none of this would ever start to take place. So just to get a general, understanding of what, we're talking about here, this is a mushroom. This is the cap is composed of the uppermost part of the mushroom. The fruit body is everything that's pretty much above ground. The stem is what holds the cap above ground, and the gills are underneath the cap. The gills are what sporulate. They release spore. The spore are microscopic, usually from 1 to 30 microns, and they are spread throughout the forest by wind. There's beetles insert their eggs into mushrooms, and the larvae hatch, and the grubs eat the mushroom and develop into beetles. The beetles are covered with spores. They go burrow into the trees. They plant their eggs, or they eat the wood in there. No, I'm getting this backwards. They eat the mushrooms. They go plant their eggs in the trees. They leave their eggs. The mushroom spores then colonize the tree, and the hatching larvae eat the mushrooms, the fungi that are growing within the tree. And then woodpeckers come, and there's a whole circle of just spiraling out of control into nature. So, and on the bottom we have what is, a very small part of the fruit body, or the fruiting part. We have the mycelium. Mycelium is actually the largest part of the mushroom. It grows underground and in rotten logs. The mycelium is the larger part of the mushroom. The mushroom itself is the fruiting body. So if you compare it to an apple tree, you have a whole root system, you have a trunk, you have branches and stems and leaves and the buds flower, and they get pollinated, and the pollinated bud turns into an apple. That's very much the same with a mushroom. The mushroom is a very small part of a larger network that grows underground out of view. So just a general, the life cycle of the mushroom. The mushroom grows as a fruit. It sporulates. It releases its spores. And I'm not going to go into all this corner right here because it's kind of foggy in my mind. I'm not, like I said, I'm not a biologist or a mycologist. But it goes through its diploid and haploid stage, which has to do with how many cells are involved with the spore. And then the spore actually comes in contact with the viable medium, and it begins to germinate. Spores grow into mycelium. The mycelium, when it comes in contact with another mycelium that is it's got the DNA of some other parent, it can join together, and the hyphae fuse. And the hyphae is basically mycelium that's underground. There's a lot more technical side of that, but essentially we can refer to hyphae as interchangeable with mycelium. So the mycelium kind of combines for mating, and then it grows into the dikaryotic mycelium, which has the 2 nuclei, which can then fruit again. So that's kind of the life cycle. And I believe this is the last slide I have, so we will go back into, actually growing some mushrooms. Would you see if you could turn the lights back on?
Want the lights?
Yeah, all the lights back on. And I'm going to set a timer for myself because I tend to, this is the first time I've taught this workshop. I just want to try to make sure I don't go too far over. We do need to get out of here pretty much after the hour that it's supposed to be, so that we can respect the library's operating hours. So today we're going to be working with oyster mushroom, which scientific name for oyster mushroom. This is my cheat sheet, by the way. And if you order the oyster mushroom spawn, then you'll get one of these if you get it from Fungi Perfecti. Otherwise, it's probably available online. But the oyster mushroom, the scientific name is Pleotris ostriatus. And this is kind of the box that I received from Fungi Perfecti, which is a company run by a gentleman named Paul Stamets. He's the author of this book called Mycelium Running, as well as a few other books and several others that he's co-author of. But that book in particular is a really interesting read. If you're interested in mushroom, I highly recommend it. Wow, that wouldn't go the right amount of time. So what this is myceliated wood dust. Basically the shavings or the sawdust that comes from carpentry. Of course, it's not from like particle board or anything like this. This is a hardwood, and it's a hardwood of wood that does not have a high sap content. So there's particular species of wood that different mushrooms kind of gravitate to and grow on. So there's several considerations to take into account whenever you're dealing with this type of stuff. So what I'm going to be doing today, I don't know why I jumped in and opened that yet, but that's all right. We're going to be putting this into a larger amount of growing medium, and we're going to multiply our mycelium. And we're going to put it into a bag. Jeez, what's going on with my phone? All right, maybe I'm not going to turn the timer off. I'll just keep an eye on that clock back there. Old school. Yes. So what we're going to do is we're going to myceliate a larger mass, and then we're going to put it into a fruiting stage. So when we myceliate the mass, we need to do a few things. We need to make sure that the medium that we're going to use is sterilized. So what that involves, there's a few ways to sterilize things. You can put it in hot water above 150 degrees for an hour. And I've done exactly that using this ice chest right here. I boiled water earlier. I put a bunch of boiling water into the ice chest, and I put our growing medium inside the water. And I had it opened earlier, but it was closed. It's still pretty hot. So hopefully it's not too hot for me to be able to work with a little bit later on. When you have sterilized your growing medium, you also want to make sure that the surface that you're working with is relatively sterile. So I'm going to be putting on gloves today. This little manual suggests that you wash your hands and then you sterilize your hands with rubbing alcohol. Another way that you can sterilize your growing medium is to soak it in hydrogen peroxide solution for 24 hours. That's what's suggested in this manual. I've seen other suggestions of 30 minutes or an hour. An hour seems to be pretty standard. Yes?
Can't you sterilize in the oven?
I would not suggest sterilizing it in an oven because the growing medium let's talk about the growing medium that we're going to use. So the suggested growing medium is hay or straw. Unfortunately, I could not get my hands on any hay or straw because the minimum order to get some from a farm was a little bit more expensive than what this particular program allowed me to spend on materials. So I'm going to be using cellulose, which comes in the form of newspaper, and I've seen several people online who have been growing mushrooms on cellulose. So it's not suggested by Paul Stamets. But it is something that I have seen done. There's some folks up in the Bronx that are doing it. So that's what we're going to be trying today. We're also going to be throwing in a little bit of coffee grinds. Coffee grinds are a wonderful medium for mushroom to be grown on as well. When you brew your coffee, you're percolating hot water through it, so you're essentially sterilizing it already. So what you want to do is just make sure that the coffee grinds that you used are either fresh from the same day, or if they're not, then you freeze them and you just let them thaw out at least 24 hours before you use them. The mushrooms, they need a good operating temperature between 55 and 75 degrees in order to, get their mycelium to start multiplying, or as it's called in the book, get the mycelium running. And then you need to keep it moist and a little bit humid in order for it to continue that growth. And then when you expose it to the sunlight, that's when it will start to fruit. Now it doesn't need direct sunlight, it needs indirect sunlight. It could also be in a room with artificial lighting. Direct artificial lighting would also work in order to promote fruiting. Elevated temperatures are also desired in order to promote fruiting. So anywhere from 65 to 90 or 95 degrees. You don't want to get it too hot, and you don't want to get it too cold.
How long do you have to sterilize it for? In 100 boiling water?
Just 1 hour. 1 hour. And it doesn't have to be boiling the entire time. As I did, I didn't put newspaper into the pot. I put a few pots of boiling water in with the newspaper, and then I let it sit. And it's been longer than an hour, but I'm hoping that it cools off a little bit.
Well, do you know what the eggs are made of? You know, are they...
I believe that most of these are soy inks. Most newspapers are made of soy inks. That's why they get all over your hands.
No, they used to be worse.
Yeah.
Much worse. I don't know what they did.
Right. I'm not really sure. But I just used the Post. I used about 8 New York Posts. That's one resource that the library has lots of. There's a stack of the Posts and the Financial Times and a couple other newspapers downstairs that I had that was available. So it's a nice resource, and it's free for me essentially. And anybody that can do a little bit of scavenging can get that for free as well. So what we're going to do is we're going to use our milk crate to inoculate our spawn in. And I've got, I think I'm going to put this. This is what I did earlier. I opened a trash bag. So this is a brand new trash bag. I just cut the bottom so that I could open it up and use it here on the table. Before I start pushing too much stuff, I'm going to go ahead and just put some gloves on. It does recommend that you sterilize your hands using rubbing alcohol. I'm just going to use some sterilized gloves. Now, this spawn was created in sterile conditions, so it needs as much sterile conditions as it can get in order to successfully make it reproduce and fruit. Spawn that's been created in non-sterile conditions or outside, you don't have to be so careful with because it's already kind of immune to other bacteria and fungi. So what we're trying to avoid is having all of our material already have a bunch of other mushroom spores on it, or any bacteria or any viruses or anything that could outcompete the fungi that we're trying to produce, which is the edible oyster mushroom.
What particular type of oyster mushroom is it?
This is, it's just it's a white oyster mushroom, but the scientific name is the Pleurotus ostriatus.
And
I'm not Latin.
blue mushroom.
There is a blue mushroom and a
king
brown brave,
yeah,
beige, and
pink.
king, and the pink, and the pinks are supposedly really great in warmer environments. These are supposed to be some of the most tolerant of the broadest range of environments. So that's why Paul Stamets recommends them and sells so many of them.
Right.
So I'm going to pull my ice chest around. In this case, it's not an ice chest. It's a warm chest. So that's pretty hot, still pretty hot. I'm just going to start layering some right in here. What I did was I cut a piece of cardboard and kind of put it in so that it wouldn't drip through too much. And actually, this needs to be squeezed out a little bit. It doesn't need to go in soaking wet. So I'm just going to take a few clumps out at a time, kind of squeeze it out.
Could you just soak it as a bundle, you know, like?
Yeah, you could soak it as a bundle and then break it up later on. What I did was I ripped it into shreds and then soaked it like that. It still is, it's pretty matted up, so I still need to break it up a little bit.
You couldn't put it flat like as it is?
You could, but then you have a situation where it's kind of compacted and you don't want it to be too compacted because you want the spawn to be able to kind of get into the nooks and crannies, if you will. Yeah, I should have boiled this a little bit early. It's still really hot. But that's all right. It reminds me of when I used to work at a restaurant and make mozzarella.
Oh, wow.
Yeah. That can be a hot process as well. So I'm just going to put about 1 inch of this down on the bottom, and then I'm going to put a layer of the spawning.
Does the ink affect the mushrooms at all? Or chemical also?
It shouldn't. It's a soy-based ink, so it shouldn't really affect the mushroom. And this is pretty. It's kind of. It's not solid, but it's kind of. It's all clumped together. That's what mycelium does. And that's how you know if you have a really strong run of mycelium, if it's kind of hard to break up the clumps. So it's actually a good thing. The harder it is to break up, the better the mycelium has been running in your medium. Just going to dump a little bit of this in here. And this bag should be mixed with about 5 times what this is a 1 gallon bag, and it can inoculate about 5 gallons of material.
So can you show us the ratio that you have in there with that?
Yes. Can anybody see this? I'm going to put just a little bit more around the corners. So it's just a little layer over the top a little bit, and then we'll put another layer on here. And actually, I have some coffee grinds that I'm going to add to this as well, just to mix things up a little bit. And I have a scoop somewhere. So these coffee grinds come from Starbucks. Just about any coffee shop probably give you their coffee grinds. You need to go ask for them a little before you need them. It includes coffee bags, which is fine. Most coffee bags are unbleached and not dyed in any way. So that's perfectly fine to put some coffee bags in there. It's very similar to the newspaper, as a matter of fact. And all this stuff was from earlier today. I just picked this up just a little while ago, as a matter of fact.
Is there any concern about the chemical processes for processing the beans, like any residue that would be in the coffee grinds?
Nope, I don't believe so. Now, again, I'm going to repeat, I'm not a mycologist. I do not grow edible mushrooms for a living, but if it's coffee and you're drinking it anyway, and And mushrooms actually have this amazing ability to like break down really nasty things. Not everything, I'm not going to make the claim that they can break down everything, but they are being touted as micro-remediators, if you will.
Well, Steinway uses these mushrooms, the oyster mushrooms, for bioremediation.
Yes.
Yeah.
And yeah, so there's really not that much concern over, especially what would be in the coffee, because it is something that you're already consuming. I would personally be more concerned about what's in the newspaper, but I don't think that it's anything detrimental either.
Bad news.
Bad news, exactly. So there are other ways to inoculate mushrooms. Oyster mushrooms do particularly well on smaller pieces of debris. Some mushrooms, the primary decomposers, can be inoculated directly into fallen logs. It's suggested that you don't inoculate logs immediately after they are felled or after they fall. It's good to give them a couple of weeks, but not longer than 2 or 3 months to inoculate them. And what is done is something similar to this is created, but instead of inoculating wood shavings, they inoculate little wood dowels, the little cylinders. And those are then taken, you drill a hole, or multiple holes, into your downed log, and you hammer the inoculated dowels into the log. And then you cover it with the food-grade wax, so that nothing else gets in there and outcompetes the mushroom that you're trying to inoculate. So again, I'm just taking, I have a layer of newspaper, a layer of inoculant, a layer of coffee, and I'm going to just repeat the process again.
Is there a particular kind of tree that mushrooms prefer for that?
Yes, there are a particular kind of tree. Different mushrooms prefer different trees. I have heard that shiitake is actually Japanese for oak. I don't speak Japanese, and I haven't confirmed that with anyone that does speak Japanese, but it sounds true enough. Shiitake mushrooms do prefer oak trees. Different types of mushrooms prefer different types of trees. Referencing experts like Paul Stamets and other mushroom growers to determine what type of mushroom to grow on your type of tree, or to determine what type of tree to get for whichever type of mushroom you prefer to eat, would be advisable. Does anybody forage for mushrooms? Yes? You kind of have to know your stuff in order to do that, don't you? Foraging for mushrooms is a great idea if you know your stuff. But if you don't know how to identify mushrooms, it is not a good idea. Many mushrooms are poisonous, and some of them even look like good old edible mushrooms, but are in fact can be poisonous. Yes?
Yeah, every spring I notice my neighbor has mushrooms growing on the front, the front area, by the side, closer to the street. There are mushrooms growing there. Do you think they're How can I find out it's theoretical? They come up every spring, they look wonderful. And I'm assuming they're organic, since I don't think any chemicals have been put on the grass.
Yeah, well,
I'm tempted to.
you know, there are field guides. I would suggest that you find, check out the New York City Mycology Society. They have field trips, I believe, every Monday where they go out into the wild and they identify mushrooms.
So New York City Mycology?
Yeah, I think it's the Mycological Society.
Mycological?
Yeah, and I'm not sure if it's the New York City Mycological Society. Oh, the New York Mycological Society.
New York.
Okay, yeah, the New York Mycological Society, full of very intelligent folks. They really know their stuff. They can tell you all about the diploid, haploid stages of sporulation. They can tell you all about, you know, the many different types of mushrooms. I went to an interesting talk the other day. There were some members who had started a business, and they're creating packaging containers out of mushrooms. Not food grade yet, but packaging containers nonetheless. And I thought that was extremely interesting. So there's lots of potential with fungi, which is one of the reasons I wanted to teach this class. Even though I'm not an expert, I wanted to do some research, and I knew that saying that I was going to teach a class would force me to do the research. I'm also hoping to do some informal micro-remediation of a small plot of land in Queens, not too far from here, and see if we can't get some mycelium running on that site and decrease the amount of contaminants that are inside the soil.
What kind of stuff is it?
That's a good question. Probably one I should leave for an expert, but if I were to venture a guess, or just to kind of try to remember what I read in this book, it actually is fairly good at taking out some VOCs, which are compounds in soils that can be left by industrial processes, industrial practices. There's lots of different ones, PCBs, PAHs. Most people know them by their acronyms, and they have really long words like polyhydroaromatic fluorocarbons and stuff like that.
Gasoline?
What's that?
Gasoline?
DSLE?
No, gasoline.
Oh, gasoline. Yeah, gasoline is, there's, even more than gasoline, just straight up crude oil. There have been mushrooms that have been shown to actually break down plastics, which is a form of petrol. So, where am I here? I'm going to put some more of this in. So, to me right now, the hardest part about this is squeezing out the really hot newspaper. But this right here actually is kind of a process to create. There are ways of creating this spawn without ordering it from laboratories. I am not completely comfortable doing that yet. I haven't ever really tried it, but anybody can do a little bit of research and figure it out for themselves. And it would actually be, I would encourage that, I mean, the worst that could happen is that you would not succeed. But essentially what you would want to do if you wanted to try and create your own mushroom spawn is to get the mushrooms that you want to grow. And mushrooms have different types of sporulation. A lot of the types that we eat have gills on the bottom sides, like I was showing in that slide earlier. Some of them actually have pores, like morels, they have pores, and they sporulate a little bit differently. But figuring out how the type of mushroom sporulates and trying to collect the spores is what you would want to do if you wanted to create your own mushroom spawn. Now, with mushrooms that have gills, you can put the mushrooms on a piece of paper, and just the mushroom cap, you can take the stem off and put the mushroom cap flat on the piece of paper and leave it there for 12 hours. You're going to create what's called a spawn print, and it's going to leave its mark in spores. So underneath the cap, as the mushroom matures, it drops its spores. So the longer that a mushroom is sitting in the forest floor, the more spores are released, and the thinner that cap gets. So you want to try and find the mushroom before it has dropped most of its spores. And you can tell if it's dropped a bunch of its spore or not if it's still slightly rounded on the bottom. When it starts to flatten out, then it's reaching maturity. And some mushrooms, not all of them, they start to edge up on the sides, and you can usually tell if it's done that, then it's probably released most of its spores already. But when you try to collect mushrooms to get the spore, you can make your spore print on a piece of paper, and actually it's suggested to use a piece of cardboard, because the cardboard is a good growing medium for the mycelium. Corrugated cardboard, not that thin stuff that the pizza boxes are made out of. The corrugated cardboard, the mycelium starts to colonize on it. You can keep it kind of moist and damp, and the mycelium will actually start to grow on it. If you have the spore from several different mushrooms, the mycelium will start to connect, and the DNA sets from different mushrooms will start to reproduce, and it'll create viable mushroom, or mycelium that will go on to produce viable mushroom. Once you see the mycelium growing on your cardboard, which looks, when you take a look at this, you can see that the outside it's kind of white. You'll see it a little bit more pronounced when it grows on cardboard. It has this dendritic pattern, and it looks a little bit like white mold, but a very pronounced kind of vein-like dendritic pattern. So once you see that, you can take your cardboard once it's colonized, and you can place it on a larger piece of cardboard, up to 20 times the size. And what you want to do is you want to peel apart the corrugated cardboard and place it kind of in the middle, and then put it back together. You can also do this with stem buds. So when you collect mushrooms, the bottom part of the mushroom that was underground, it has little hyphae, or mycelium, kind of attached to it. And the hyphae actually come up through the stem and into the caps, and then the ends of the hyphae is what releases the spores. Those hyphae down on the bottom are, to compare it to something from the plant kingdom, it's like taking root cuttings. So you can take the stem buds, and you can also put them in between two layers of corrugated cardboard, and keep that moist, not wet, but keep it damp and keep it in a warm environment. And that hyphae will start to run, the mycelium starts to run. And then, again, when you see that start to happen, you can start reproducing this by putting it in a larger piece of cardboard. And then once you have a nice piece, you can either use it as an underlayment for a bed of mulch, or you can use it by putting it on top of a bed of mulch. Another interesting idea that's being promoted in this book, Mycelium Running, is to use bags made out of burlap. So either jute or hemp bags that have been untreated to grow mycelium in. So filling that with wood chips or wood shavings or even coffee grinds, and then placing that on top of the cardboard to create your mycelium bag. And that's where the mycelium would start to grow. So I'm going to do one more layer of this, and then we'll move on. And I think we're going to go ahead and try to Maybe we don't have enough time. We'll see. Oh no, maybe not. We may do a bunker, we may start a bunker bag tonight.
Is that something that you could have outside, or is that?
This is definitely something that you could have outside. Like I was saying earlier, it does not reproduce, or the mycelium will not run if it's under 55 degrees. So definitely later in the season, or next season, if you will, this can be put outside, but for now it needs to remain in a sheltered location indoors where the temperature is going to be correct for it to actually start growing. So I'm going to be cutting it a little bit close. Does anybody have any questions that they would like to start asking? Not that I can guarantee to answer all of them, but I'd like to give you an opportunity to start, just to lob stuff out there. Yes?
How long would that soil, that combination would last as far as usage?
As usage?
Yeah.
Well, it's going to fruit in 1 to 3 months, depending on the temperature and the moisture levels. You'll start seeing mycelium kind of colonizing the outsides of this. We're going to keep it in a bag. We're going to allow the bag we're going to poke holes in the bag so that it can breathe. Mushrooms do breathe oxygen and release carbon dioxide just like we do. They need light, not direct sunlight, but light, and they need water. And they don't need it to be wet water. They need it to kind of be a humid, moisture in the air kind of water. But this will last probably 9 to 16 months. That's going to be when it starts to fruit, and you're going to be able to pick the fruit. And then after that, as I was saying, mushrooms are decomposers. What you have left in here after that 9 to 16 months is really great mushroom compost, so that the usable life, if you will, continues on. And we'll be transforming these newspapers and coffee grinds and inoculated wood shavings into edible mushrooms and really great soil that we can use in our gardens.
What's the best way to keep it moist? Spray or?
A spray bottle is probably the best way to keep it moist. I've heard of people putting misters on timers in really, kind of high-tech operations. But for this, we're going to be keeping this at the library, actually. And I'm going to be misting it on a regular basis. And I'm just going to use a regular mister. One important thing to note about that is the water that you use. Mycelium is a very fragile living thing, and our tap water contains fluorine, or fluoride, which, be it as it may, I don't know if any of you are for or against fluorinated water, but you need to make sure that the fluorinated water does not get applied to mushrooms. Or if you keep worms at home, or even, if you're starting seedlings, it's better to use rainwater or water that you leave out in a container, an open container overnight in order to let the fluorine evaporate before you use it in anything that is a resemblance of a natural process. Now, human biological systems are natural processes. We are somewhat more dynamic. I'm not going to argue for or against fluorinated water, but apparently it's okay for us. I wouldn't suggest it for your mushrooms or any smaller invertebrates or even your seeds and to some degree plants.
So, I'm sorry. The fruiting would appear the way the examples in that photograph are along the sides of the
Yes.
crate?
Yes. That's how the fruit would appear. That it would grow out the sides just like that.
So once it starts fruiting, that would be continuous for, you say, up from 9 to 16 months?
Yes.
That would do it?
Yeah, you can harvest this. Let me keep this on real quick. We don't have very much time left. There's only about 5 minutes left. But yes, we can. Once you get your first harvest, there's usually a second harvest. It's a little bit smaller. Sometimes, depending on what sort of material you have, what the growing conditions have been, you could possibly get a third harvest out of this. So it's this bag costs about $15. So we're inoculating a good chunk of material here. What I wanted to show you real quick before we wrap up was the hydrogen peroxide method. So what I have here is a 2-gallon bucket of water. It doesn't have, it's not completely full, it's to about right here. What I want to do is, and I wasn't planning on having coffee grinds, but now I have a dirty container, but that's all right because we're going to be inoculating coffee anyway. You want to get regular hydrogen peroxide, the kind that this is full grade. This is 3%. This is kind of the standard of what's sold in the stores. It's not you said food grade.
This is the food grade, which is 35%.
No, that's not. We want to dilute this to 0.00%. So this is what this equation right here is. We have a desired 0.00%, and what we have is 0.3%. So I'm kind of giving this in increments so that you can kind of measure it out, because the way that he describes it and the way that he's saying to grow these is in a 5-gallon bucket. So he actually, his example was 5 gallons. I figured I'd just do 1 gallon so that if you have a 1-gallon bucket or a 2-gallon bucket or a 5-gallon bucket, you can just multiply that up. So 128 ounces is in 1 gallon. And then when you do all that math, which I did with the calculator earlier, I'm also not a math genius, you end up with 1.28 ounces. Now that's not a whole lot. So I'm just going to kind of eyeball this. It's not going to be exact science, but I'm going to put about 1, 2, we're multiplying that by 2, so it's roughly 2.5 ounces that we're looking for. About 2.5 ounces right there. And this is a rice bag, you get your rice in your bags. And I'm going to make a bunker bag out of this later on. This needs to soak for an hour at least. If we take Paul Stamets' advice, we'll do it for 24 hours, which I have time to do that. So we'll do it I'm going to do this for 24 hours. And then I'll take this bag out. And what I'm going to do is I'm going to take, probably not all of this, I'm going to take enough coffee grinds to fill that bag up. Well, I'm going to mix it with this and fill that bag up with it. And then I'm going to keep that in a warm, moist location and mist it often. And it should also grow mushrooms.
They grow out of the bag?
They grow right out of the bag. There's, the jute or the hemp fiber is relatively loose, so there's gaps in there that it can grow out of. The mycelium will start to run inside the bag, but it'll also discolor the bag. It'll start to colonize the bag itself because it is natural material, the jute or the hemp. So it'll colonize the bag itself, and the mushrooms will grow directly out of it.
Do you need
So, in other
newspaper?
words, you can't reuse the bag for another.
No, the bag will become
Right.
that compost.
Exactly.
You know, the bunker bag system is primarily meant to kind of. Paul Stamets is recommending it for remediation of sites or sites where you have toxic runoff, like CAFOs, where they keep a bunch of livestock and you have effluent from that livestock. You put bunker bags inoculated with these mushrooms. It captures the runoff from these livestock, and it actually cleans it before it hits the watershed.
The toxic sludge. that
Exactly.
kind of stuff.
Exactly.
Did you say you mix mycelium with coffee grinds and then?
Yes, I'll mix the coffee and the mycelium together, and then put it into the bag. Yes.
That reminds me of the way they're using oysters to clean the water.
Right.
This thing.
Yeah, I mean, oysters it's a completely different process. They're bivalves. They take in the water and filter it, and they spit out the clean water. Mushrooms, the hyphae, go into the soil. They secrete acids, and that breaks down compounds into smaller digestible pieces. It absorbs those, and it takes it where it needs to go. If you go way back in the biological process, I mean, we have a similar system in our stomach where we digest things with stomach acids. So in that way, we're somewhat related.
Mushroom.
To fungus, yes.
So when you put it into the bumper bag, you're not going to put newspaper?
You can, but I'm not. You can do it with coffee grinds or newspaper or wood chips. I have plenty of coffee grinds that just go straight coffee grinds. I didn't want to do straight coffee grinds in this because it could have gotten messy as we got towards the top. So we're running out of time, but did everyone have a chance to sign the sign-in sheet? Okay. So I'm going to give away a few of these. These are made from a wonderful company out in Oakland, California. They're mushroom. It's a little mushroom garden. Inside this is an inoculated bag of coffee grinds. You can take this front panel off, cut open the bag, and soak it overnight or for 12 hours in, again, non-chlorinated water, rainwater or water that's been left standing, or in a pinch you can boil water. But you don't want to put it in hot boiling water. Then you put it back in the box. The instructions are all in here. I trust that all of you can kind of go with it. Keep it in a warm spot and mist it often. So how many names do we have there? What number did we get up to?
Some people didn't sign in.
Yeah, if you didn't sign in, then you're not going to be...
I counted more people than names.
What's the name of the company?
This is Back to the Roots, is the company that makes this. It's two guys in business school together. One of their professors said that you can grow mushrooms on spent coffee grounds. So they started growing these mushrooms. They pretty much turned down high-paying jobs to start growing mushrooms professionally. And they stopped growing mushrooms and started producing these things to let other people grow mushrooms.
They made over $1 million in 1 year.
Yes, it's kind of amazing. All right,
School.
who's number 9? What do we got down on number 9? I'm going to pull a few of these out. This one goes to number 9. And
Number 9 is me.
there you
I
go.
wanted one.
Yeah, there you go. What number are we up to? We don't have a 34, do we? No, we're only on 16. All right, 34, 35, 25. Geez, I thought, okay. Getting closer. I have way too many of these numbers in here. 15. Let's see, 15 is Susan Sims.
Are you kidding?
Nope.
Everything in my life.
And Shirley Chai. Number 15. All right, let's see. Number 2.
We're almost finished.
Is G. Fieldstad. That's you? Oh, Jay. Number 16 is the last person that put their signature on here.
It must be me.
Congratulations, guys. Well enjoyed. If you would, please take pictures. You know, we'd love to see how your mushroom garden progresses. We'd love to write a little post about it on the Greening Queens Library blog, which can be found at greening.queenslibrary.org. Strongly consider it. Check that out. Lots of fun going on here at the library. I hope that y'all can join us in the future for more programming. Greening.queenslibrary.org. queenslibrary.org. That also has a calendar of our events that are going to be coming up. If you put your email address on this, you'll also get, just every once in a while, a little update of what we're doing. We had e-waste recycling days. We had Shred Fest the other day. We shredded personal papers so that people wouldn't get identity theft, and we recycled all of it. It would have been awesome if we could have inoculated all of it. But I thank you all for coming out, and I look forward to seeing you all.
Inoculate me.